Hi, and welcome to Mr. Fisher's Biology Podcasts. This podcast deals with the origins of life. So we begin our journey with Aristotle. And back in 384 to 322 BC, Aristotle was the first person to give a general idea of how living things could arise. And he proposed the idea of spontaneous generation. Now, we also call spontaneous generation abiogenesis. Spontaneous generation or abiogenesis is the idea that living things come from non-living matter. Now, what's important to realize is that with Aristotle's idea, it lasted for something like 2,000 years. People believed in the idea that living things came from non-living matter. So the next couple slides are some examples. So for people, for 2,000 years, it was very obvious to understand that muddy soil gave rise to frogs. They noticed that every spring, frogs would emerge from the soil, and so it was very clear that the mud was giving rise to the frogs. So a non-living thing was giving rise to a living thing. Another example, this is a classic one, that if you leave meat out in the sun all day, that the meat would produce flies. So it was perfectly understandable back then to believe that rotting meat gave birth to flies. And people believed this, remember, for 2,000 years, until people began to look at trying to disprove spontaneous generation. So there were some people that questioned the idea of spontaneous generation. The first of them, Francesco Redi. And Francesco Redi performed an experiment. I guess it's important to note here that prior to this, experimentation wasn't done to show that spontaneous generation was the case. It was simply just observation without experimentation. So Francesco Redi was the first person to undergo an experiment. And he provided very significant evidence against spontaneous generation. So his experiment involved 3 glass jars, the first of those being unsealed, the second of those being completely sealed up, and the third one was sealed with a wire gauze. And what he noticed was that in the unsealed flask, there were maggots on the meat. In the sealed flask, though, there were no maggots on the meat. So spontaneous generation suggested that the meat was producing the flies. And what Redi showed was that the meat is not producing the flies. And in fact, when he put the gauze on it, that there were flies on the gauze, there were some maggots on the gauze, but there was none on the meat. So this was the start of disproving spontaneous generation. So what Redi sort of concluded was that spontaneous generation was not the case for larger organisms. Other organisms were giving rise to the maggots. So flies were giving rise to the maggots. So people began to believe this idea that living things or larger living things were giving rise to other living things. It still didn't explain microscopic organisms like algae or bacteria, and so people believed that they were still spontaneously produced from nothing. And enters another scientist and the theory finally changes. So this is Louis Pasteur, and everyone knows Louis Pasteur from pasteurization. Louis Pasteur developed a series of apparatus that we still use today, just in more advanced forms. But here's what Pasteur believed. Pasteur believed that microbes come from the cells of organisms carried on dust particles in the air, not the air itself. So Pasteur created special S-shaped flasks, and he put broth into those flasks. And each flask was boiled and placed at various locations. Now, the S-shaped flask itself was intended to trap any dust particles containing bacteria. So this is the S-shaped flask, and right here at this S neck here is where dust would be collected, and nothing would get into the broth. So that was the idea behind the S-shaped flask. He left them at various locations. He noticed that the broth itself didn't turn cloudy. He could not find any microbes, and he did notice that dust collected in the neck here. So here's an idea of his experiment. He boiled the flasks to kill off any microbes that would be present there. And over time, he noticed that there was no growth of bacteria. On some of the flasks, he broke the stems. And so he allowed air to get into the flask. And what he noticed was we got microbacterial growth. Pasteur's conclusions were that the S-shaped flask kept the microbes out. It still allowed for air to come in. And so, in other words, it proved that microbes can only come from other microbes. Life comes from life, and we have the idea of biogenesis. So biogenesis, by definition, is that living organisms come from only other living organisms. So spontaneous generation is completely disproven, and we move into an era called biogenesis. And this is really the start of the science of biology. So biology begins at biogenesis. It still didn't explain how first life originated, and that's the next part of this podcast that we're going to look at. So we're going to start with a fellow by the name of Alexander Oparin back in 1930. And Oparin hypothesized, he didn't really test this, but he hypothesized that energy from ultraviolet lightning and volcanoes cause chemical reactions in the atmosphere which form small organic particles. And those particles would precipitate into the ocean, and first life came from oceans. Now remember, he didn't actually test this. This was a hypothesis, but these would have been sort of what was found on early Earth. There would have been a lot of lightning and a lot of volcanic activity as the Earth was first forming. Now this new life would use the organic molecules as food. So these guys were called heterotrophs, meaning that they eat other organisms, or they obtain their food from the environment. They don't make their own food. So Oparin's hypothesis was known as the heterotroph hypothesis. So the first life was heterotrophs. This was confirmed by two scientists by the name of Stanley Miller and Harold Urey. And they supported his theory, but they went ahead and they tested it. They created this apparatus with gases that would be thought to be in the atmosphere. So methane, ammonia, water vapor, and hydrogen were all placed into this area right here. They added an electric spark, which would have simulated lightning. And what they found down here in the trap is that this brown sludge containing amino acids, sugars, and other small organic molecules began to accumulate. And these, of course, we know are the building blocks for life. Amino acids are the precursor to proteins, and sugars would provide energy. And there were other smaller molecules as well, some ATPs and some very early nucleic acids. So all of this can be formed just from the conditions of Earth. And this was shown in the the Miller-Urey experiment. Next step was to form complex molecules. And so it is known that when you have amino acids and heat, and you don't need oxygen present, amino acids will bind to form proteins. Now this takes a lot longer. We have enzymes in our body that do this a lot quicker. And it was thought that these molecules, these early proteins, were formed in small warm pools of water, not in the ocean, but on land in small warm pools of water. Now in 1992, another neat experiment, a guy by the name of Sidney Fox produced protocells. Protocells are large ordered structures with membranes that could grow and divide. They're not living though. And these were formed with conditions that would have been found at the time on those, or was believed in those small warm pools of water. So where were the first cells? The first cells were formed about 3.5 billion years ago, according to the fossil record. They were prokaryotic. meaning that they had no nucleus. And they likely evolved from these protocells, and they were anaerobic, meaning that they don't use oxygen. They fed on organic molecules, so they were heterotrophic. They don't make their own food. Later, autotrophic organisms that could have arisen had an advantage because they could now produce their own food. And the very first photosynthetic organisms began to form. With photosynthetic organisms forming, they produced oxygen in our environment, into our atmosphere, and aerobic organisms began to evolve. And aerobic respiration, remember, is way more efficient than anaerobic respiration. So the origin of eukaryotes began with the idea known as the endosymbiont theory, that there were prokaryotic cells and they were invaded by other prokaryotes. So these were things like mitochondria and chloroplasts. They were early prokaryotes because they both have their own set of DNA. They're produced from each other. And they formed rather a mutual relationship with these larger prokaryotic cells. And so this diagram really just shows what's happening. We have these ancestral prokaryotes. This one would be an early mitochondria, would produce ATP itself, and these ones here, early chloroplasts, they would produce their own sugars. And this would be our early prokaryote, our larger one, and began to engulf them. And rather than just consuming them, those mitochondria began to evolve themselves inside of the prokaryote, and we get the formation of early eukaryotic cells. And now we come to multicellular life. Multicellular life has evolved in the past 600 million years, and all organisms have really evolved from here. So that's our podcast on the origins of life.